So yes, we are done with both the phases. One is photochemical phase, second is biosynthetic phase. Two phases are done, right ma? And then we have also dealt about photorespiration, that is C2 cycle. What is that we are left over from photosynthesis? Factors affecting photosynthesis. After this we will be starting respiration in plants. So what are the factors that are deciding how much photosynthesis should happen or not? Okay, it can be a positive effect or it can be a negative effect. These factors can be both extrinsic factors and intrinsic factors. Extrinsic and intrinsic. It can be external factors or internal factors. External factors include three factors beta, which includes light, temperature, percentage of carbon dioxide, amount of carbon dioxide that is present, and water. Okay, these are the external factors which will decide the rate of photosynthesis. Intrinsic factors will include the number of leaves that are present, what are different types of leaves that are present, okay, amount of photosynthetic products that are present, okay, age of the plant. These are what? Intrinsic or internal factors which affect the photosynthesis. So, in any biochemical process it might be, many factors are affecting that biochemical process, means amongst all that factors, which factor will decide the biochemical reaction? The factor which is close to the minimal value will decide the biochemical reaction. I repeat, in any biochemical reaction, if there are multiple factors that are affecting the process, okay, which factor will decide the rate? The factor which is close to the minimal value. That was proved by Blackman's law of limiting factor. Once look into the theory also beta. Photosynthesis is the process by which the plants transform light energy to chemical energy, that is why it is known as physico-chemical process. It is under the influence of several factors, both external and internal. Blackman's law of limiting factors is observed when several factors affect any biochemical reaction. It was put forward by F. Blackman, who is a British plant physiologist. He said that if a chemical process is affected by more than one factor, remember it is affecting by more than one factor, then its rate will be determined by the factor that is nearest to the minimal value. It is the factor that directly affects the process of its quantity is changed. Okay? For example, amongst the external factors we will take, amongst all these external factors, which is very minimal? CO2. It is just 0.03% in the atmosphere. So, this percentage of carbon dioxide majorly will decide the quantity of photosynthesis. Okay, amongst all the factors, which is the deciding factor? Carbon dioxide. Why? It is close to the minimal value. Who said that? Blackman's law of limiting factor. Clear, right ma everybody? Now, these are the external factors we have discussed. First is light. Okay, light quality, duration of light and light intensity. These are the three ways where light will be affecting photosynthesis. What are the three ways? Quality, intensity and duration. Okay, amongst this, light intensity plays major role. Rate of photosynthesis initially, look at this graph, very important, 1 or 2 times asked in your PYQs. So, in this graph if you observe, light intensity is plotted at x-axis and rate of photosynthesis at y-axis. Okay. Here, as light intensity gets increased, rate of photosynthesis also increases. Okay. Once after it gets saturated, it reaches to the saturation point where maximum rate of photosynthesis occurs just at the 10% of total sunlight. Okay. Then though you increase the light intensity, there will not be any increase in rate of photosynthesis. It comes to a saturation point. At what point of light intensity photosynthesis rate comes to saturation? 10% of total sunlight only. Only 10% of total sunlight will make the rate of photosynthesis come to a saturation point. Okay. Look at the theory also once. Rate of photosynthesis increases as light intensity increases until it reaches to the saturation point. Saturation occurs at 10% of full sunlight. Especially at what regions? Blue and red wavelengths of optimum photosynthesis. Light duration does not affect the rate of photosynthesis, but it affects the process, like how much photosynthesis is happening. Okay. Light duration has a special effect where you will learn it under photoperiodism, short day plants and long day plants. Okay. So, till here though it is clear, no, what are the major points that you have to remember? With the increase in light intensity, rate of photosynthesis increases, it comes to a saturation point and then there will not be any increase. Okay. 10% of total sunlight, it comes to a saturation point. Clear, right? Any doubts anywhere, ma? Yeah. And then about carbon dioxide concentration. We know what percent of carbon dioxide is present? 0.03 to 0.04 in the atmosphere, and carbon dioxide is the most limiting factor that is present. And till this increases to 0.05 also, the rate of photosynthesis increases from 0.03 to 0.05, rate of photosynthesis increases. Beyond 0.05, it leads to toxicity and it will affect the process of photosynthesis. Okay. What is the most limiting percentage? 0.05%. Clear? Any doubt anywhere, ma? Here, how C3 species and C4 plants, both C3 and C4 plants respond to carbon dioxide concentration. Okay. The intracellular carbon dioxide concentration is measured as microliter per liter. Okay. This is the measure of intracellular carbon dioxide concentration. Remember that. So, here first at low light intensities, when light intensity is low, at low light intensities, though you increase carbon dioxide concentration, there will not be any effect. Okay. Carbon dioxide concentration, even if it is high, at low light intensities there is no effect. The effect happens only at higher light intensities. At high light intensities, if carbon dioxide concentration is increasing, then it comes to a saturation point. Beyond that, there will not be any increase of photosynthesis. What are those saturation points at higher light intensities? Carbon dioxide concentration and rate of carbon dioxide uptake. This is the graph. This graph is not there in your NCERT, not required to remember, but for your understanding I have put. So, if you see for C3 species, it is around 450 microliters per liter. This is the saturation point. For C4 plants, can you see here? Yeah. For C4 plants, it is 360 microliters per liter. Can anybody tell me why C3 plants have more saturation point than C4 plants, beta? Sit straight everyone, listen attentively. Why C3 plants have more saturation point than C4? Because 6 carbon molecules. Give complete explanation. You are saying the first word and stopping. Why? In C3 plants, what is the enzyme that fixes first carbon dioxide? Rubisco will perform both carboxygenase activity, carboxylase and oxygenase activity is performed. So, there is a competition between carbon dioxide and oxygen, no? Yes or no? Both are present at equal concentration, then there is no problem. But if oxygen concentration increased at higher light intensity, what happens? Carbon dioxide gets solubilized. whereby eventually oxygen concentration increases than carbon dioxide. Okay. So that is the reason eventually oxygen concentration increases. That is the reason more carbon dioxide requirement will be there for saturation point. Understood everybody? Should I repeat it once again? Yeah. Rubisco will have both carboxylase and oxygenase activity. Okay. So when carbon dioxide and oxygen are at the equal ratio, there is no problem. I said you at higher light intensities there will be an effect in carbon dioxide concentration. When light intensity increases amongst these two, which gets easily solubilized? When carbon dioxide gets easily solubilized, eventually oxygen percentage increases or not? So to meet that, we have to increase carbon dioxide concentration or not? Yes or no? That is the reason the saturation point of carbon dioxide for C3 plants is high. This explanation is required for you to answer statement based questions. Okay. Second point, if you see in C4 plants, what is the primary carbon dioxide fixation enzyme in C4 plants? Pepcase. Pepcase will bind only to carbon dioxide. Okay. Phosphoenol pyruvate carboxylase, it binds only to carbon dioxide. So there is no problem, right? Understood the role of carbon dioxide everybody? Light intensity is clear to you, carbon dioxide effect is clear to you. Saturation points very important, Nana. Okay, just to remember, C3 plants it is around 450 microliters per liter. C4 plants it is around 360 microliters per liter. Clear, right? Understood everyone? Yeah. And then water. Okay. So the role of water comes into picture both in flaccid conditions and in turgid conditions. Okay, in flaccid conditions what happens? The loss of water from plant is due to what? Transpiration. Okay, then first step, any problem happens in the house, what do? First is we shut the doors. Okay, same the case with the plants, it will close the stomata, it will shut its door. Then it prevents the exchange of gases like carbon dioxide. This reduces surface area, reduces metabolic activity inside the plant, thereby you will observe what? Wilted leaves. Okay. So with water decrease or water increase, rate of photosynthesis is affected. This much you remember, it is enough. And then is temperature. Okay. Temperature plays a crucial role. Okay. More than light reactions, with the increase in temperature, the enzymes that are present in dark reactions are more sensitive. Like pepcase and rubisco, they are more sensitive to higher temperatures. Like if you observe, the light reactions are temperature sensitive, but they are affected to a much lesser extent. The dark reactions being enzymatic, they are completely what? Temperature controlled. The C4 plants respond to higher temperatures as they show high rate of photosynthesis. C3 plants have much lower temperature optimum, around 20 to 25 degrees Celsius. C4 plants are tropical plants whose temperature optimum is around 30 to 45 degrees Celsius. Clear, right? Next is oxygen. So the role of oxygen, Warburg effect is not given in your NCERT, but we may expect a question. What it says that at higher concentration of oxygen, what does rubisco do? It performs oxygenase activity. Okay, rather than carboxylase activity, it will perform oxygenase activity. So higher concentrations is affecting the rate of photosynthesis. This phenomena is known as what effect? Warburg effect, as it is discovered by him. Okay, this prevents rubisco in binding with carbon dioxide, hence carbon dioxide fixation is affected, thereby rate of photosynthesis is reduced. You know the process, just address the name, enough. Okay, so you are clear with external factors. First, what did we learn, ma? Light intensity. Second, carbon dioxide concentration. 0.03% in the atmosphere is the normal concentration. Till it increases to 0.05, there is no problem, but beyond that, it affects the rate of photosynthesis. And in C3 plants, if you observe, what is the saturation point? Saturation level? of carbon 450 microliters per liter. In C3 plants, at low light intensities, does rise or fall in carbon dioxide concentration show any effect? No. At high light intensities, it shows the effect. Next, water, high water concentrations or low water will affect the rate of photosynthesis. Oxygen concentration will also affect the rate of photosynthesis, which is explained under Warburg effect. Next is about internal factors. Directly I will finish off here only. Internal factors will affect rate of photosynthesis. It is on many criteria. It is based on number or arrangement of mesophyll cells, orientation of leaves, age of leaves, size of leaves, number of leaves, amount of chlorophyll and photosynthetic products. What are photosynthetic products? Starch or glucose. Okay. If it is already abundantly present inside a cell, is there any requirement for photosynthesis to occur? What does this products do? These products will inhibit the primary step of the reaction, which is known as what? Feedback inhibition. Not just here anywhere. If products of the biosynthetic pathway, any biosynthetic pathway are already present in a cell, then what those products will do? Those products will inhibit the initial step of the reaction, inhibit the initial enzymatic step of the reaction, thereby the process doesn't occur. You can see it here. Okay. Glucose, if it is already formed, it will affect the carbon dioxide fixation. Clear? Understood? These are the external and internal factors. Done with this, ma? Now open your NCERT. In that, factors affecting photosynthesis. Only single page it is there, right? Read it in my presence. If you are having any doubt, you just ask me. Now let's begin with respiration in plants. What are the concepts we have to cover from here? First is you have to know about what is respiration, then about different types of respiratory substrates that are utilized for respiration. What is cellular respiration? Types of respiration based on the requirement of oxygen or in the absence of oxygen, or based on the type of respiratory substrate utilized for respiration. Two types of classifications you have to learn, two types based on respiratory substrate, and second is based on presence or absence of oxygen. Next is glycolysis, followed by anaerobic respiration. You will learn about two types of fermentations. One is lactic acid fermentation, second one is alcoholic fermentation, followed by oxidative decarboxylation, Krebs cycle and ETS in aerobic respiration. Aerobic respiration. Okay, and then amphibolic pathway, last is respiratory quotient. So first, if you have to know what is respiration, tell me what is the primary difference between breathing and respiration? A common question. What is the difference between breathing and respiration? Inhaling and exhaling is known as breathing. Okay. What is respiration? Breakdown of complex molecules into simpler forms. For what reason? For no reason it is breaking. For providing energy. Okay, for providing energy. Okay, breakdown of complex molecules, that is complex biomacromolecules, into simpler forms, thereby releasing the energy, required energy for cellular metabolic activities to occur. That is known as what? Respiration. So does breathing occur in plants? We know we do breathe by nostrils, but do plants breathe? That is the primary question. Do plants breathe? Yes, plants do breathe. Plants do breathe. If plants breathe, why are complex respiratory organs absent in the plant? Three major reasons. One thing, when we have learnt anatomy of flowering plants, we learnt that most of the plant body is composed of what tissue? Parenchymatous tissue. Parenchymatous tissue is loosely arranged or tightly arranged? Loosely arranged with intercellular spaces. So, each plant part will not show a greater demand for respiratory needs. Every plant part will take care of its own gaseous exchange, and gases are easily diffused because majorly it is of what? Parenchymatous tissue. If you take leaves, in leaves what happens? All the tissues are at the surface area. So, they take care of their own respiratory exchange. In stem, if you observe, there is an organized layer formation of epidermis, endodermis, and then about pericycle and vascular bundles. In all those organized layers, the tissue is parenchymatous tissue. Same the case with roots. Okay. So, as it is abundantly composed of parenchymatous tissue, gaseous exchange happens because of diffusion, and in leaves the tissues are closely adjoined to the surface area. Okay. So, in stems, for the gaseous exchange they take the support of lenticels, open pores which are present on the stems, and in the leaf it is stomata. Okay, for gaseous exchange, stomata and lenticels. Clear everybody? Any doubts anywhere? So, do complex respiratory organs present in the plants? No. How do they take care of breathing? Via stomata and lenticels. That's it. Simple, right? And then, so now in chloroplast of a cell, assume this as a plant cell. Okay, assume this as a plant cell. Here, chloroplast which is present inside the plant cell. Inside chloroplast, where is glucose formed? Stroma of the chloroplast. This glucose, if it has to get stored inside the chloroplast or in the cytoplasm, it will be stored in the form of starch. If it has to be transported for respiration or oxidation, it will get transported in the form of sucrose via phloem to different parts of the plant body. And this sucrose, once it reaches to any cell, the same cell I have drawn, let us suppose this sucrose has entered to any other cell. The sucrose is a disaccharide, no? Yes. When it breaks into two monosaccharides. What are they? Glucose and fructose. Okay. Glucose via glycolysis in cytoplasm, it will provide an end product. What is that? Pyruvic acid. After 10-step long process, we will learn about glycolysis right now. Okay. First you get a framework what is happening inside the cell. Glucose will first turn to what? Pyruvic acid. This process is common in any cell, be it aerobic respiration or anaerobic respiration. This process is common everywhere. Pyruvic acid then enters to the matrix of mitochondria. This is outer mitochondrial membrane, this is inner mitochondrial membrane or cristae, this is intermembrane space, and this is mitochondrial matrix. Okay. So, pyruvic acid will enter into the mitochondrial matrix, and then second step occurs. What is that? Oxidative decarboxylation, followed by TCA cycle in mitochondrial matrix, and in the inner mitochondrial membrane, that is cristae, what happens? Electron transport system, which is the last step of aerobic respiration. Clear till here? Understood, right? So, now you have understood the common pathway, like what is happening inside plant cell. Glucose formed, if it is stored in what form? Starch. Transported? Sucrose. Sucrose enters into any cell, what is the first activity done? It gets break to glucose and fructose. What does glucose do? Glycolysis. Forms an end product in cytoplasm of the cell, that is pyruvic acid. Pyruvic acid immediately enters into matrix of mitochondria. Second step is oxidative decarboxylation, and then TCA cycle, and then in inner mitochondrial membrane it is electron transport system. Clear? Done everyone? Now we will see different types of respirations. Types of respiration. You can take down types based on what happened? Respiration, what is breathing? Okay, how do plants breathe? Via stomata and lenticels. Only 2 points we have discussed. This you hold on, then immediately put the title types of respiration. Okay. Based on respiratory substrate, and then second is based on presence or absence of oxygen. Based on respiratory substrate, we have two types of respirations. One is floating respiration, and second one protoplasmic respiration. Floating respiration, the respiratory substrate will be fats or carbohydrates. Protoplasmic respiration, it is proteins. Based on presence or absence of oxygen, we have two types: aerobic respiration and anaerobic respiration. In both the respiratory pathways, one thing is common, that is glycolysis. Followed by glycolysis, here two things can happen. One is alcoholic fermentation Second is lactic acid fermentation. If it is turning to aerobic respiration, inside the mitochondrial matrix 3 steps will happen. First is oxidative decarboxylation, followed by TCA cycle, followed by ETS. Now you got a clear framework. Just note down what is respiration, what are types of respiration, that's it. Do plants breathe from that area? If you are clear on the NCERT statements, it is enough. Done, ma? And now first we will learn what is glycolysis, and then we will move on to anaerobic respiration. Glycolysis is also addressed as EMP pathway, which is discovered after 3 scientists who are Emden, Meyerhoff and Parnas. Emden, Meyerhoff and Parnas. Be here all, be here. Okay. Glycolysis, you will get a definite question, many at times asked. Okay. Glycolysis is also known as EMP pathway, which is named after the scientists Emden, Meyerhoff and Parnas. It is a 10-step long process. First 5 steps are known as activation phase, next 5 steps are addressed as energy extraction phase or payoff phase. The name itself is saying in the first 5 steps what happen, energy is taken by the cycle. In the next 5 steps what is happening, it is giving off the energy. In that you have to remember each step, what is the enzyme involved in each step. So first step, once glucose entered into the cytoplasm, it gets first phosphorylated to glucose 6-phosphate by the enzyme hexokinase, for this phosphorylation of glucose it requires one ATP. This ATP is taken from the cytoplasm itself. From this ATP it has triphosphate, right, adenosine triphosphate. One phosphate is utilized for the formation of glucose 6-phosphate. ADP is being released out into the cytoplasm. Fine, right? This is the first step. Clear everybody with the first step? Next. Second step, glucose 6-phosphate to fructose 6-phosphate. This is an isomerization reaction by phosphohexoisomerase. And third step is known as rate limiting step. This is catalyzed by the enzyme phosphofructokinase, which is rate limiting step and phase maker enzyme. Why is it called like that? This is addressed as phase maker enzyme, and this step is known as rate limiting step because this single step will decide on how fast or slow this cycle should happen. Okay, fine, understood. So for this step also, as kinase is involved, see here also kinase enzyme will do what? Phosphorylation activity. Okay, so here phosphofructokinase, from fructose 6-phosphate to fructose 1,6-bisphosphate, means at first carbon and 6th carbon phosphate group is attached. Fine. So at first step and third step what are utilized? ATP is utilized. Next, fourth step, fructose 1,6-bisphosphate, which is 6 carbon, is cleaved to 3 carbon compounds, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This is by the enzyme aldolase. Okay, and these are 3 carbon, right? They are isomerized to each other, okay, because from 6th step glyceraldehyde 3-phosphate will run the show. Once G3P is done to 1-pyruvic acid, then DHAP will isomerize to G3P and it will again run the next 5 steps, okay. So it will do in what? 2 cycles, but we are considering for 1 glucose. So what we will assume? 2 glyceraldehyde 3-phosphate. At the end what is formed? 2-pyruvic acid, because we are assuming for 1 glucose. Understood the concept? First step is clear, by hexokinase glucose to glucose 6-phosphate by hexokinase, ATP is utilized. Second step, glucose 6-phosphate to fructose 6-phosphate by phosphohexoisomerase. Fructose 6 to fructose 1,6-bisphosphate, phosphate group attached to first carbon and 6th carbon by what enzyme? Phosphofructokinase. Fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and DHAP, as it is cleaved, it is by what? Aldolase enzyme. 6th step here by isomerization, right? They are changing their structure, number of carbon is same. So here this is by isomerase. This is glyceraldehyde 3-phosphate dehydrogenase, okay, because NAD plus is reduced to NADH plus H plus. So at 6th step what is released, ma? NADH plus H plus is released into the cytoplasm. This is by glyceraldehyde 3-phosphate dehydrogenase. Then 7th step, 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid, okay. Here one ATP is released into the cytoplasm, then phosphoglyceric acid to 2-phosphoglyceric acid by what? Kinase, phosphoglycerokinase and phosphoglyceromutase. Here it is phosphoglycerokinase and this is by the enzyme phosphoglyceromutase. Here what is that phosphorylating? ATP. ADP is taking phosphate group from where? From substrate. ADP is taking phosphate group from substrate, that is 1,3-bisphosphoglyceric acid. Look at here, 1,3-bisphosphoglyceric acid, that is from first carbon phosphate group is being taken. Then it is turned to 3-phosphoglyceric acid. Clear, right? This is known as substrate level phosphorylation. Okay. 8th step is phosphoglyceromutase, where 2-phosphoglyceric acid is formed. Then 9th step is catalyzed by the enzyme enolase, which is dehydration. 10th step, from phosphoenol pyruvate to pyruvic acid, which is again a substrate level phosphorylation. ATP is released into the cytoplasm. Can anybody tell me what is substrate level phosphorylation? What is substrate level phosphorylation? Here ATP is released, yes or no? ADP is taken from cytoplasm. It requires one more phosphate for the formation of ATP. That phosphate is taken from this substrate. That is the reason it is known as substrate level phosphorylation. Okay. Same the case here. 10th step is catalyzed by what? Pyruvate kinase, phosphorylating ATP. ADP and inorganic phosphate is taken from what? PEP, phosphoenol pyruvate. Understood 10 steps? Any doubt anywhere? Okay. Once we will revise this by taking down the energetics of glycolysis. Phosphofructokinase, whose doubt it is? It is known as pacemaker enzyme because it will decide the accuracy of glycolysis in a cell. For example, you are on the fasting days. Okay, you are on the fasting days. If high amount of glycolysis happens, what happen? You will get you will fall sick soon. Okay, glucose is very much slow present in your cell, okay, and high amount of glycolysis happens, what happens, you will fall sick soon. Okay, you ate huge, so much of glucose is present in your cell, at that time what happens, high amount of glycolysis happens. So what is that step which is deciding the accuracy of glycolysis? Third step, which is known as what? Rate limiting step. By what enzyme? Phase maker enzyme, that is phosphofructokinase. Understood? Any other doubt from this? Yeah. Next here you see ma, bioenergetics. First, in first and third steps, ATP is utilized. How much? 1 ATP at first step, 1 ATP at third step. So total 2 ATP are utilized by glycolysis. At sixth step, NADH is released into the cytoplasm. Yes or no? At sixth step, it is released into cytoplasm. How much we are considering for 1 glucose? So it is 2 NADH. 2 NADH. At 7th and 10th step, 2 ATP at each step are released into the cytoplasm. Yes or no? 2 ATP are released into the cytoplasm at 7th step. And 10th step also 2 ATP are released. So total how much? 4 ATP. Okay, this 4 ATP which is directly released into the cytoplasm is known as gross ATP. Okay, 1 NADH is equal to 3 ATP, right ma? 1 NADH is equal to 3 ATP, 1 FADH2, 2 ATP. Okay, if you have to know accurately, so it is 2.5, it is 1.5. For the ease of calculations, at this level we will consider it as 3 ATP and 2 ATP. So here 2 NADH are released at 6th step, which means 2 into 3 total how much? 6 ATP. 6 ATP plus 4, how much ma? 10 ATP is the total ATP that is released. From this 10, 2 are utilized, right, at first and third step. So it is how much? 8 ATP. Okay, and net ATP is 2 ATP. From the gross ATP which is released, 4 ATP is the gross ATP, right, directly released into the cytoplasm. From that direct release, 2 ATP are utilized at first and third steps. So the net ATP is 2 ATP. Net ATP, how much? 2 ATP. Total ATP is 8, gross is 4, net is 2. How much amount of NADH is produced? 2. At which step? 6th step. Understood? Look at each step, each enzyme and the energy. Okay. If you are having any doubts, just ask me. Understand glycolysis totally. Let us suppose a prokaryotic cell. Listen here everybody. Glycolysis is clear, no? Now, let us suppose a prokaryotic cell or a eukaryotic cell, to muscle cell. Okay? Any cell where anaerobic respiration is happening. Now, inside a cell cytoplasm, glucose is turned to pyruvic acid. Okay? If it is running out anaerobic respiration, pyruvic acid inside cytoplasm only, it will choose either of the pathways. One is alcoholic fermentation, second is lactic acid fermentation. Okay ma, clear? One is alcoholic fermentation, second is lactic acid fermentation. Alcoholic fermentation, if you observe, glucose to pyruvic acid. At sixth step, remember this sixth step clearly, NADH is released into the cytoplasm, right? NADH is released into the cytoplasm. This NADH is utilized for alcoholic fermentation. First, pyruvic acid will turn to acetaldehyde, which is decarboxylation by the enzyme pyruvic acid decarboxylase. Just two steps are here, note down the enzymes, enough, okay? Pyruvic acid decarboxylase. Fine. 2 molecules of pyruvate to 2 molecules of acetaldehyde. Okay. Decarboxylation, this is 3 carbon, this is 2 carbon. So, 2 carbon dioxides will be released into where? Cytoplasm. And then acetaldehyde to ethanol. Okay, by alcoholic dehydrogenase. Fine. So, at this step, the NADH which is released at the 6th step of glycolysis is being utilized, and this turns to what? This gets oxidized to NAD plus, which is again utilized at which step? 6th step of glycolysis. Clear? Any doubts anywhere ma? So, here first what happens? Glucose to pyruvate under glycolysis, you know. At 6th step NADH is released. Okay. Pyruvate to acetaldehyde, pyruvic acid decarboxylase to carbon dioxide. Then acetaldehyde turns to what? Ethanol. Pyruvic acid decarboxylase will do the decarboxylation activity. Alcoholic dehydrogenase will form the product, that is ethanol. Clear till here? Any doubts anywhere? Alcoholic fermentation is clear, right? So, here you have to note down 2 major points. Alcoholic fermentation will be utilized by the bacteria which are doing fermentation, like for example unicellular fungi yeast for the preparation of duff. Okay. And second, the cells will get toxic with the accumulation of 13% of alcohol in a cell. Leads to what? Toxicity. 13% of alcohol in a cell will lead to what? Toxicity. Clear? Any doubts anywhere? Second, about lactic acid fermentation. Unlike alcoholic fermentation where we have learnt it under 2 steps, in lactic acid fermentation there is only single step where pyruvate is 3 carbon, lactic acid is also 3 carbon. Okay. Here it is catalyzed by the enzyme lactic acid dehydrogenase. And the cofactor that is present to catalyze this is zinc. Okay. Here it is magnesium is the cofactor. Okay. For pyruvic acid decarboxylase, what is cofactor ma? Magnesium. For lactic acid dehydrogenase, what is the cofactor? Zinc. Okay. And here also same the case that is happening in the glycolysis at 6th step, NADH is released, that is utilized for lactic acid fermentation which regenerates back NAD plus, which is utilized at 6th step of glycolysis. Clear? Any doubts anywhere? No. You are taking down? Anybody? Glycolysis you are taking down? No, right? This also. So, next we will directly move on to aerobic respiration. If anybody is taking down, I will wait. If not, I will move. So, as we have learnt in the beginning, after glycolysis, when pyruvic acid is formed, this directly enters to which place of mitochondria? Mitochondrial matrix. In mitochondrial matrix, before TCA cycle, one step happens. What is that? Oxidative decarboxylation. Second step, that is oxidative decarboxylation. Where is the site of this reaction? Matrix of mitochondria. Listen carefully, everybody. Listen carefully. Pyruvic acid along with the coenzyme A along with NAD+. I have taken for single pyruvic acid, but in the energy sheet you have to remember it is for one glucose molecule. Okay. So, NAD+ turns to what? Acetyl-CoA. The reaction itself is with decarboxylation. This is how many carbon beta? This is forms CO2 and NADH. NADH plus H plus. If it is for 2 molecules of pyruvic acid, 2 CoA are required, 2 NAD plus are required, 2 acetyl-CoA, 2 carbon dioxides are released, 2 NADH plus, 2 H plus. Okay. So, at the end of oxidative decarboxylation, what is that it is formed? Acetyl-CoA and 2 CO2, and how much amount of energy is released? 2 NADH. Okay. Here the enzyme that is present is pyruvic acid decarboxylase. As it is performing the activity of decarboxylation of this substrate. We can also consider the enzyme pyruvic acid dehydrogenase. In the options, any of these are correct. If any of these options are given for you for oxidative decarboxylation, it is correct. Why dehydrogenase? Because NAD plus will be turned to NADH. Clear with this step, second step? Any doubts anywhere, ma? Anybody can ask. Okay. Next. TCA cycle. Okay, ma'am, no problem. TCA cycle is tricarboxylic acid cycle, also known as Krebs cycle. Citric acid cycle also. Here, the first substrates that are adjoining is what? Acetyl-CoA. Immediately after oxidative decarboxylation, it binds to oxaloacetic acid. Oxaloacetic acid is how many carbon beta? 4 carbons. Oxaloacetic acid is 4 carbon. Acetyl-CoA is 2 carbon. Total how much? 6 carbons. By the enzyme CoA it forms and the enzyme citrate synthase. By the enzyme citrate synthase it forms citric acid, which is also known as citrate, which is how many carbon? 6 carbon. 2 carbon coenzyme A, acetyl CoA, and 4 carbon oxaloacetic acid. By the enzyme citrate synthase, it forms 6 carbon citric acid, which is also known as citrate. Now, one hydration reaction and one dehydration. Water molecule is taken and lost. By the enzyme aconitase, this forms 6 carbon isocitrate. One more substrate will happen in between, that is 6 aconitase, not required. Okay, so by aconitase what substrate is formed? Isocitrate, which is also 6 carbon. Isocitrate will undergo decarboxylation. This step is important. Wherever I am keeping a star mark, have an additional focus and look into it. So, the 6 carbon isocitrate is turned to what? 5 carbon alpha ketoglutarate by the enzyme isocitrate dehydrogenase, okay, where here NAD+ is reduced to NADH. If I have to take a single TCA cycle, you tell me at this step how much NADH is released? How much NADH? 1. If I have to consider 1 glucose, 2 NADH. Only at this single step I am asking. How many NADH are released? 2. Alpha ketoglutarate, which is 5 carbon, is again decarboxylated and this forms 4 carbon succinyl coenzyme A by the enzyme alpha ketoglutarate dehydrogenase, where NAD+ is reduced to NADH. Okay. Succinyl coenzyme A is 4 carbon, right? This turns to the substrate succinate. Okay. Here again substrate level phosphorylation happens, that is SLP, where GDP will be taking the inorganic phosphate from this substrate and this turns to what? GTP. Okay. It can be GDP or it can be ADP. Any of the nitrogen base can be present and it can form GTP, guanine triphosphate or adenosine triphosphate. Succinate to fumarate, which is 4 carbon, it FAD reduced to FADH2 by the enzyme succinic dehydrogenase, then fumarate to malate by the enzyme fumarase, malate to oxaloacetic acid by the enzyme malate dehydrogenase. Here again NADH is released. You have to remember these spots specially wherever NADH and FADH2 formed, especially at this substrate level phosphorylation also. At this step where alpha ketoglutarate is reducing, at this step succinyl CoA undergoing substrate level phosphorylation, succinate to fumarate, where succinic dehydrogenase is the complex 2, okay, and then from malate to oxaloacetic acid. This step. Clear? Any doubts anywhere, ma? No, right? Now if you look into the bioenergetics of TCA cycle, only TCA cycle. Of single TCA cycle, and if you consider from 1 glucose, 2 TCA cycles should happen, no? You tell me, for single TCA cycle, how much NADH is released? First this row, you tell me how many NADH for single TCA cycle? 1, 2, 3. 3 NADH. How many FADH2 are released? 1. How many ATP? 2 TCA cycles. 6 NADH. Done? Second point, if the question is asked in mitochondrial matrix, okay, in mitochondrial matrix. Just a second. Okay. Second is in mitochondrial matrix. In mitochondrial matrix, two events are happening. One is oxidative decarboxylation, second is TCA cycle. In oxidative decarboxylation, if we have to consider single oxidative decarboxylation, how much energy is released? 1 NADH. But 2 pyruvic acids are there for 1 glucose molecule. 2 oxidative decarboxylations, how much energy? 2 NADH. See, in the next class, okay? Energy sheet. Cheppu. Say Jyotika. For 1 glucose molecule. 1 glucose only. Only one hero. Our hero is glucose here. No double action. Only single action. So the energy sheet of aerobic respiration, if you observe, first glycolysis, then oxidative decarboxylation, third is TCA cycle or Krebs cycle, okay. So here if you observe, how much ATP is released? Out of 4 ATP, that is gross ATP, 2 are utilized. So we are considering the net ATP, that is 2 ATP plus 2 NADH. Okay. From oxidative decarboxylation for 1 glucose I told, means 2 pyruvic acids. How much NADH? 2 NADH. In TCA cycle, remember, wherever decarboxylation occurred, there NADH is released. First 2 steps. Okay. So here from isocitrate to alpha ketoglutarate, 1 step where NADH is released. 6 carbon, 5 carbon. 5 carbon alpha ketoglutarate to succinyl coenzyme A, which is 4 carbon. Okay. Here one more NADH is released. First 2 decarboxylations, and then at last step of Krebs cycle, that is from malate to oxaloacetic acid. At 3 spots NADH are released. Only at 1 spot FADH2 is released. That is complex 2 of ETS. Okay. From succinate to fumarate, okay, and one substrate level phosphorylation from succinyl coenzyme A to succinate, where ATP is released. Now got some clarity at least. Third listening, you people only will tell me. So here for 1 glucose, 2 TCA cycles, how much? 6 NADH. How much FADH2? Now, so in the beginning of the class, I think I have not told you about cellular respiration. Cellular respiration is also the same, oxidation of macromolecules inside a cell and release of the energy in the form of ATP. Why ATP is known as cellular energy? Why not NADH or FADH2? Very good. ATP is the simplest form of energy, and the cell's metabolic pathway will be using the ATP directly for whichever metabolic reactions it requires. To run the cellular metabolism, it will use the direct energy in the form of ATP. So, all these major forms of energy should convert into the simplest form of energy, which happens in ETS. Okay. So, in ETS, single NADH converts to 3 ATP, 1 FADH2 to 2 ATP. So, here if you consider only ATP, here it is 2 plus 2 into 3, 6 ATP plus 2 into 3, 6 ATP, 6 into 3 plus 2 into 2, total how much ATP? Calculate properly and tell me. 38 ATP you will get. If you say 36, that is wrong. 38 ATP is the total ATP that is formed in aerobic respiration. See, if there is a question in your exam, what is the total ATP that is synthesized or released from aerobic respiration, your answer should be 38 ATP. If they are asking you what is the total amount of ATP that is released via aerobic respiration in a eukaryotic cell, your answer should be 36 ATP. Why? No. It is because, listen, in glycolysis 2 NADH are produced, no, that NADH will be converted to ATP in the last step only. Okay. For that NADH from cytoplasm, it has to get transported to mitochondrial matrix. To transport NADH into the mitochondrial matrix, there is a shuttle system that is operated, which is known as glyceraldehyde 3-phosphate shuttle system. Okay. To transport 1 NADH, it will use 1 ATP. So, to transport 2 NADH, how much ATP it is using? 2 ATP. Okay. So, from 38 eliminate 2 in a eukaryotic cell, what will be the answer? 36 ATP. That is all. Understood everyone? Any doubts anywhere? So, this is the complete energy sheet ma. Okay. So, this is the complete energy sheet. In our next class, we will be discussing about electron transport system. We will be discussing about respiratory quotient and about amphibolic pathway, and then we will discuss all the NEET previous year questions from photosynthesis and respiration, all PYQs. Come prepared with these 2 chapters for next class.